very much. I'm Peter Heifetz, I'm the founder of Therapeutics. Thank you all for coming this morning, and looking forward to kicking off this RESI conference. So Therapeutics is developing an inhaled enzyme treatment for obstructive airway diseases and a few other things, as I'll describe. We are in the JLabs accelerator in San Diego. Been there a little bit over 5 years. We raised a $2.3 million Series A, and then about $2.7 million in non-dilutive funding. Our technology was in-licensed from National Jewish in Denver, with a little bit from Novartis. This had passed through with me before being separated from Novartis when they spun off their life sciences way back, and then we've taken this and improved it. In addition to the original IP from National Jewish, we've just gotten new patents issued based on our modified molecules. We've got a pretty long runway on IP. Our team there has worked together in a number of different capacities. So cystic fibrosis is our lead indication. It's the most prevalent fatal inherited disease. And it's a disease with a single gene defect, and that results in defective or obstructive mucus in the airway, which is no longer able to be transported. And it's this static mucus that leads to the symptoms in the disease of obstruction, infection, inflammation. Those are the things that kill you. And typically with cystic fibrosis, patients are on multiple treatments, as many as 6 different therapies designed to restore mucus clearance. There's some new therapies from Vertex Pharma, which are essentially rotor-rooters. The primary gene defect is an ion transporter. That results in loss of chloride and bicarbonate secretion in the airway, and then that is in turn what drives the mucus defects. So our strategy is to restore normal mucus transport. If this is successful, it should be a disease-modifying therapy preventing progression to those serious symptoms. It is based on thyrodoxin, which is a naturally secreted enzyme on the airway. And thyrodoxin is a selective disulfide bond reductase, and it has properties that I'll describe in a minute, which are very relevant for this. And we think there is a loss of the activity of thyrodoxin as a consequence of the CFTR defect. What separates this from a chemical reducing agent is that it has selectivity, which I'll describe in a minute. This means it only targets the most abnormal disulfide bonds rather than non-selectively taking things apart. It is a genotype-independent therapy. It will work on patients with any mutation in cystic fibrosis, and we believe it's going to work alongside drugs like Vertex's to improve all of those patients. And then about 20% of patients are not candidates for CFTR modulator therapy, and this might potentially be a first-line therapy. So thyrodoxin is the most important secreted human protein you've never heard of. It has essential roles inside the cell, which is one of the main reasons why it's never really been developed as a drug before. It is a subunit of ribonucleotide reductase. There's no nucleic acid biosynthesis without it, so you can't progress past a single cell without this protein functioning. And there are a variety of other intracellular functions involving transcription factor activation. Extracellularly, it's a different molecule. It acts homeostatically on mucosal surfaces in the body. And its selectivity means that it will only target 2 to 3 of the 20 possible structures of disulfide bonds, and that's mediated by the shape of its active site pocket. As an enzyme, it has catalytic activity and very high potency compared to small molecule thiol agents. And then it has a very unusual pH response. It's active across a broad range of pH because of hydrogen bonding at its conserved active site pocket. So these are the 3 characteristics which really separate this from thiol agents, reducing agents, which have been used for many years, pretty much unsuccessfully in this disease. So the 3 activities of it are relevant to cystic fibrosis. It normalizes defective airway mucus and actually restores normal mucus transport. I'll show you some of that data. It reduces inflammation by damping down responsive T cells to stimuli, and this is a fairly broad activity. It acts at the level of mediated release all the way through ICAM and LFE1 interaction. And it acts to activate a class of secreted antimicrobials called defensins. When these are in the oxidized form, they have very low activity. When reduced selectively by thyrodoxin, they become very potent, including with activity against Pseudomonas aeruginosa, the main pathogen in cystic fibrosis. So you can't just deliver thyrodoxin because of the intracellular effects. And so what we've done then is engineered the molecule to make it an extracellular activity. It will find the compatible disulfide bond, break the bond, and remain bound to one of the 2 cysteines. And this means that the drug is now locked on its target. It can't be taken up intracellularly. We get excellent safety, but It also increases activity because we now shift the equilibrium of open disulfide bonds away from oxidized intact bonds. And so we have about 400 times greater activity, maybe 1,000 times greater activity than acetylcysteine, approved drug for treating mucus. It retains the selectivity and pH independence of thyrodoxin. And then, as you see in the graph here, this is shows on the left the binding on the mucus layer. At the top is an active version of a molecule, ORP100S. The bottom is an inactive control. In the middle, it shows it restores flow to mucus by gravity. And then on the right, this is a micro-optical coherence tomography image on a tracheal surface. The bright blob on the bottom is a human CF sputum on a trachea treated with saline. On the top is with the drug and saline. And so it's able to drill into this mucus and relieve the excess viscoelasticity and cohesiveness. And this is reflected by the orange bar, which shows it's able to restore a fully normal rate of transport. In gray is human DNase. That's been an approved drug to degrade secreted nucleic acids. Many patients are on that, only about 20% respond. Still a $600 million a year drug for Roche. So we've significantly de-risked this program. We've scaled manufacturing up to our first half-kilo batch. It's non-glycosylated. We can make it in E. coli. We have a stable lyophilized formulation that is extremely good for keeping the molecule active in storage. We then reconstitute it for delivery. We're working with a leading manufacturer for a nebulizer. This is delivered as an aerosol to the lung. In cystic fibrosis, there is a very large market opportunity, and we were able to create some strong barriers with broadly issued patents. It was a very successful IP campaign for this, both US and international. And the mechanism is common to multiple diseases. Anything on the airway surface where you have acidification, that will inactivate the cofactor of thyrodoxin and result in loss of activity. So the rare respiratory diseases are themselves very large markets. Bronchiectasis is our secondary target for this. There are no approved therapies for that. We're also now looking at a synthetic peptide version for COPD and potentially some others. And then there's some non-respiratory indications on other mucosal surfaces in the body. Fairly standard orphan drug pathway with a single phase 3 trial. We're currently raising a convertible note to get us to a first in human, and then a bit more in our Series B that would get us through a proof of concept study in a large number of patients. Compared to other drugs in the space, we stack up extremely well. As I mentioned, we think this is going to be very complementary to the new drugs from Vertex, and it has some significant advantages over other approaches. There have been drugs which have been designed to inhibit sodium channel activity, which was one of the hypotheses for how that ion channel defect caused abnormal mucus. Those drugs have now all failed clinically. So we think we're really on to what the natural mechanism is here. So in summary, it's a large opportunity, especially when you look beyond cystic fibrosis to other obstructive diseases. Hundreds of millions of people are affected globally. It's an approach based on a natural homeostatic mechanism. Everything we've seen has been very safe. We did an acute inhalation study in rats, couldn't get a tox dose so far. We're planning on our repeat dose GLP tox later this year. IP has been successful for this. We have allowance that we issue soon in Europe and Japan. We have issued patent in the US and pending composition claims. Overall risk is very attractive for this. Really, it's the only question left is, does this work in people? And that, of course, is the question that, when you look at investing in a drug, that's the risk you want to have left. And as a biologic, we've had very good success on the CMC side. And so what we call Therapeutics technology is our optimized thyrodoxin, which we're looking for a range of opportunities. Thank you very much.
Questions? Questions are allowed.
Thank you, everyone.
Hello?
Okay, the next presenter will be Smart Farm Therapeutics from Boston. Jose?
Great. Thank you very much. Thank you for all coming early this morning, and thank you for the organizers for inviting me to speak. Biologics has transformed medicine, and as the CEO of Smart Farm, our vision is that we want to have your body produce those transformative biologics from within. We are a next generation non-viral gene therapy company developing a broad platform of DNA and RNA therapeutics that are delivered via nanoparticle-based approaches. We have a very highly experienced team to execute this that has taken drugs all the way through approval. Currently, we are raising a private Series A round, and we're seeking investment in this round that will take us to IND ready for our initial proof of concept. As many of you know, there's been excitement out there with the medical promise of gene therapy coming to fruition. As a graduate student in the 90s, I worked on some of the early adeno and AAV vectors, and it's been really exciting to see that come to some of the recent approvals for gene therapy. So this is obviously a space that we're just at the beginning, and there are many new diseases to address with this exciting therapy. A lot of you, probably the majority of what you've seen out there in respect to the recent approvals are viral vectors that are depicted on the right. And as you know, viral vectors are disabled viruses that are used to deliver the DNA, to deliver nucleic acids such as DNA or RNA. We focus on non-viral methods, which we see as the next generation of approaches for gene therapy. And these involve taking nucleic acids such as DNA or RNA and encapsulating them in nanoparticles to deliver into the cells, much like a virus does, but without a lot of the immune issues that you have with viruses. So, in many cases, viral vectors, have had issues with, or some of the challenges with viral vectors are you have immune responses that prevent re-administration. So if you don't get, if you have waning expression, it'll be very difficult to re-administer. The other issues are for vectors such as AAV, about 40 to 70% of people actually have pre-existing antibodies to many of the vectors that exist out there. And finally, as many of you may know, there's a huge bottleneck in manufacturing, and it's very expensive to manufacture these viral vectors and do it reproducibly. We focus on a platform of both RNA and DNA, although we're leading with our DNA vector technology. But this, we feel, allows us to address a wide variety of diseases with our platform. RNA has shorter expression and is more suitable for things like vaccination, tissue engineering, etc., and tissue regeneration. DNA has longer term expression and is more suitable for going after more chronic and rare diseases where you need prolonged expression. So our lead program that will be our initial proof of concept for our approach is in Gaucher disease. Gaucher disease, as many of you may know, is an inherited disease that is a deficiency in the glucocerebrosidase or GBA gene. This ends up in having macrophages store up this sphingosine, which actually causes toxicity to the macrophages. And then the macrophages collect and release cytokines in both the spleen and the liver, and this causes enlargement of the spleen and the liver, and is definitely debilitating and can be fatal, and results in low anemia, bleeding, skeletal abnormalities, and pulmonary abnormalities. The current standard of care is a protein therapy that needs to be infused every 2 weeks. So Gaucher is actually a very established disease, and specifically Gaucher type 1 is more systemic as opposed to 2 and 3, which have more neurological manifestations. Currently, the global Gaucher market is north of $1 billion, and however, it is ripe for disruption because the current standard of care, as I said, needs to be a purified, expensive protein that has to be delivered every 2 weeks. So our goal is to address this by delivering a non-viral gene therapy that will express the GBA gene systemically and stably over 3 to 6 months, even up to a year. The approach that we use is using a proprietary DNA vector that has been, that is minimally immunogenic and also has very stable expression once it's inside of a cell. This this DNA encodes the GBA gene and then is coupled with a nanoparticle of, and we have two basic delivery systems. One is a lipid nanoparticle that is suitable for delivery to the liver, and the other is a polymer-based approach that is suitable for injection into the muscle, and basically using the muscle as a factory to produce the GBA. I'm going to skip ahead for the sake of time and show you some of the data. The figure here basically demonstrates that with a lipid nanoparticle formulation, with our DNA vector expressing, in this case, a fluorescent reporter gene, this is delivered to a mouse model where you have look for expression in the liver. And on the left is the percentage of the total liver cells that are expressing the gene, the fluorescent gene. And on the right is a publication using different strains of AAV with a very similar experiment. And what you can see is with the lipid nanoparticle-based delivery of this reporter gene, you're getting efficiency of delivery of the gene to the liver on par with that of AAV. In terms of our polymer-based approach, on the left shows figures with different transgenes being delivered into the anterior tibial muscle of a mouse. But the key is the middle figure where you have a serum protein that you're measuring. In this case, it's secreted alkaline phosphatase. And you can see with a delivery into the muscle, you're getting therapeutic levels of between 10 to 20 micrograms per ml of the secreted alkaline phosphatase. Now, in terms of our milestones, as I mentioned earlier, we're currently raising a private round that will get us to key preclinical data as well as regulatory input so that we are IND ready, with the goal of having non-human primate data and a preclinical package by the end of the year, and the ultimate goal of a CTA application for a clinical trial in 2021. So as you can see, our goal is to finalize our candidate and seek regulatory approval in the next quarter with the goal of getting that IND ready package towards the end of the year. In terms of Gaucher as a lead program, this would establish proof of concept, in a systemic disease where you need expression of a therapeutic protein. But as you can imagine, once you establish that, there's a very broad array of diseases that could be addressed. And even within the lysosomal storage diseases, which Gaucher is one of those diseases, there are many other lysosomal storage diseases that have current markets and represent, high unmet need with patients that we could address once this proof of concept is established. And our pipeline is not only focused on Gaucher and enzyme replacement therapy, but we also have RNA programs for tissue regeneration, and we also have a program where we can express biologics such as cancer biologics with our DNA vector in vivo. So imagine your body being able to produce therapeutic antibodies from your muscle or systemically without having to have multiple infusions of expensive biologics. We have a very experienced team that's taken multiple drugs to approval and developed multiple drugs, as well as co-founded companies. And finally, I'd like to end with just what are our distinguishing factors. We're focused on non-viral gene therapy. We're focused on gene-encoded therapeutics, although our technology could certainly enable gene editing. We have both a broad DNA and RNA platform. We're focused on the DNA platform, but our delivery methods allow synergy between these two platforms. We have a minimally immunogenic nucleic acid delivery system and vector. We have protein and glycosylation experience, which is important for optimizing the proteins that you deliver. And many of our methods have established, for example, the DNA vector, have established GMP methods so that this bottleneck is avoided. So that's that's all for today, and I thank you for your time, and have a great meeting today. Okay, any questions? Yep.
Can you use this system to express antibacterial peptides?
Yeah, certainly if it's a peptide-based antibacterial, so any protein-based, this can easily be put in. One thing for the sake of time that I didn't mention is, as many of you know, AAV has packaging constraints in terms of the size of the gene, and whereas our vector system does not. So even if your gene was very large for your antibacterial protein, this could be fit in there. And that's evidenced also by being able to fit the heavy and light chains into our vector.
Are the nanoparticles stable through oral dosage, or it has to be intravenous?
Right now, we're focused on either intravenous for liver or localized delivery for muscle, or even it could be localized delivery into a tissue or a joint space, but we haven't really explored oral. There are some interesting technologies out there that we've looked at, and it'd be something of interest if there was a mature technology that could deliver. Yeah. Any other questions?
What would be the closest competing technology?
Well, I mean, there's obviously viral vectors, but I think as we talked about, there are some inherent issues with viral vectors. There are many companies doing RNA-based approaches, such as Moderna, so that would be kind of another approach, but I think the RNA is short-lived, so it keeps them from really addressing long-term expression. And then finally, there's some other companies using other types of DNA vectors, linear vectors, what are called doggy bone vectors that are end-sealed as well. So these could represent other vectors. For the sake of time, I don't have time to go into why we think our vector has advantages, but I think those would be some of the technologies that we would be working on. I think there's one more question in the back.
Have you done a non-human primate biodistribution study?
So with the polymer-based approach, that has been dosed to non-human primates, but a comprehensive study about where the particles localize haven't been done. This has been done in small animals. And then with the lipid-based approach, once we have a final formulation, it doesn't really make sense to use a non-optimized final formulation. We're going to do those exact studies in terms of safety and then look for distribution. But the lipid nanoparticles are fairly specific to the liver, and this is well known because there's a lot of knowledge about the lipid nanoparticles. They use the ApoE receptor. And then we have a second level of specificity in that our gene uses tissue-specific promoters so that you can avoid off-target expression. Any other questions? I think I may be out of time, but happy to... did you have another question? No? Okay. Hey, thanks a lot for your time. No, it's early, and look forward to seeing you today at the meeting. Thank you.
So the next company up is Validus Cellular Therapeutics out of Colorado. Ethan.
Thank you, it's great to be here. I'm Ethan, I'm with Validus. Slides are coming up. I want to tell you a story today about a patient who's in the ICU right now. We're going to call this patient Marie. So Marie is a 65-year-old lady who finally decided it was time to go through with that total knee replacement that she'd been putting off for years. Right? So she'd had cortisone shots, she'd done everything she could, but she had no choice. It's time to go through with the surgery. She put everything in place, arranged for recovery, and everything went as planned until Marie started to notice that she had some pain that was different than what she was experiencing with recovery. She asked her doctor about it, and with some concern, her doctor put her on antibiotics. But those antibiotics didn't solve the issue, nor did the subsequent round of slightly different antibiotics, or the inpatient infused antibiotics that she had after that. Ultimately, Marie was faced with having a 2-stage revision surgery to take out the old implant, clean the knee, and put in a new implant. In fact, she had a total of 13 surgeries, and they couldn't resolve the issue. Marie was faced with losing her leg to save her life. But that wasn't the end of the story either. Months after amputation, doctors found femoral osteomyelitis in Marie's femur, and that's now moved to her hip, and that's landed her in the ICU. These are devastating circumstances, and they're becoming more and more frequent. In 2013, Dr. Arjun Srinivasan, the Associate Director of the U.S. Centers for Disease Control and Prevention, said, "We're here. We're in the post-antibiotic era. There are patients for whom we have no therapy, and we're literally in the position of having a patient in a hospital bed with an infection for whom something we could have treated 5 years ago even, but now we can't." Are more antibiotics the answer? Not in the short term. These drugs take years to develop and produce. And there's very few drugs in the pipeline, and those that are very specific to types of organisms, and when they're most effective, they're most at risk for emerging resistance. So what if we made existing antibiotics more effective? I'm an entrepreneur and infectious disease expert, and I've co-founded a company called Validus Cellular Therapeutics based in Aurora, Colorado, just outside of Denver. And we're developing the answer for infections like Marie's. We're making a cellular therapy product called Veropicin CT, directed at antibiotic-resistant infections. Veropicin CT will be an allogenic activated mesenchymal stem cell, or MSC-based therapy that will resensitize resistant infections to antibiotics. This approach is already working in dogs at one of the world's leading innovative veterinary schools at Colorado State University. There, Dr. Stephen Dow, a veterinarian and scientist, has been faced with trying to treat animal infections without many tools. And so in his laboratory research, he's developed a program where he can activate these mesenchymal stem cells and resolve these resistant infections like you see here. On the left, you can see there's a dog with a chronic infection on its paw, something that's been treated with multiple courses of antibiotics without success. And on the right, after 21 days of activated MSC therapy along with antibiotic pressure, the infection's gone and the paw's healed. But how does this work? Well, the activated MSC therapy takes advantage of an immune modulation, primarily with M1, M2 macrophage. So out of recent research in the immunology field, people are beginning to understand more and more the importance of these two types of macrophages, especially when bacteria are present from a medical procedure. Primarily, M1 macrophages are responsible for this inflammatory response and the cleanup, whereas this M2 macrophage is really more responsible for the rebuilding process and tissue remodeling and repair. When you have a tissue injury, especially with bacteria present, the remodeling gets locked into this M1 class and you can't make a switch to M2. This activated MSC therapy promotes that transition from the M1 response to M2. Take a look at these cells from an animal infection where the immune cells were stained red if they were M1 and green if they were M2. In the control image on an animal not receiving this MSC therapy, you can see the majority of the immune cells are red or in that M1 state. This is responsible for the chronic inflammation and the non-healing response in these animals. But the activated MSC therapy, you get a majority of M2 cells, so you get that rebuilding, repair, and healing process to occur. But that's not all. These MSCs need to be able to target biofilm infections. So bacteria exist in these states of biofilm. Take a look at these surgical wires used to close a patient's sternum after open heart surgery. The presence of biofilm, which is very, very difficult to even make out on the wires, prevents the healing process to occur. And so you need something to directly target the biofilm as well. These MSCs are very good at producing antimicrobial peptides that are very effective at even killing biofilm cells. And so here's 3 different antimicrobial peptides, LL37, surfactant protein D, and lipocalin. And they're stained in each panel in a red stain. And the MSCs are blue. So you can see how much production antimicrobial peptides are in the MSC population. Validus is looking to move quickly to bring this therapy to patients. We have a strong leadership team, including myself, Dr. Stephen Dow, and 2 frontline physicians that are faced with treating patients with these chronic infections every day. We have a strong advisory team, including experienced advisors from both Colorado State University and the University of Colorado Anschutz Medical Campus in Denver. We have regulatory support, of course, because this product will need to go through an intense regulatory cycle. One of the issues with biologics and especially cell therapies is that you need to have a consistent cell source, and you need to be able to process these cells in an efficient manner. We already have these partnerships in place, taking advantage of the renowned Charles C. Gates biomanufacturing facility on the Anschutz Medical Campus in Denver, as well as Ascent Biologics, which is a recent spin out from a longtime tissue banking facility, Allosource, which is located in nearby Centennial, Colorado, just south of Denver. We'd like to move quickly, and we want to get through IND conversations with FDA this year, and we'd like to get it first in human trials in 12 months. We have everything set up to do this, except the equity piece, of course. So we're seeking $2 million to meet these goals. We think this is an attractive investment opportunity. The world of pharmaceutics is beginning to realize how important cell therapies are, and they're an untapped resource for treating many types of infections. And the regulatory climate is loosening around the stem cells and other cellular-based therapies as well. We're excited to bring this therapy to patients, and we're looking for the right equity partners to do that. And I'm happy to take any questions. Thank you. Yes?
Speak about the IP strategies and IRA.
Right, so we filed 2 years ago, the University of Colorado and Colorado State filed a PCT patent on the way the cells are activated and the way the cells are treated and the way the therapeutic is put together. So we received our first office actions on those just recently, and they look promising, and so we'll have nice patent portfolio building from that as well. Good question, thank you.
Are there any similar approaches that are close to it?
You know, there's been a lot of therapies that have used an MSC or mesenchymal stem cell based therapy. The uniqueness for Validus is that we've figured out a way to really efficiently activate these cells. So what's commonly been done on an infused mesenchymal cell therapy base is that when the cells are infused, they get trapped in the lungs because they're not activated. They're just sort of, they're stochastically put into the body and they don't go anywhere beyond the lungs. And so what we're seeing is that we actually get the infused cells to move to the sites of infection, seek out the infection and activate the immune response, directly kill the biofilm as well. Other than that, there aren't many cellular therapies really directed at infectious disease. Everything's vaccines and antibiotics, and both are difficult to develop and challenging to use. Yeah. Yes?
Have you performed any experiment on drug-resistant bacteria? What's the performance?
Yeah, so a lot of the studies that we've done are clinical dog studies and lab animal sort of rodent models. And in the rodent models, we will establish like a MRSA infection. In spontaneous infections in dogs, of course, they're all over the place, and they're things that, I'm a microbiologist, and there are things that are difficult for me to pronounce. So there's stuff that's not commonly seen, but it's typically very resistant because to enroll in the clinical dog study, the patients have to have multiple courses of antibiotics fail. And so the infections are almost always resistant. Yeah. Okay, thank you.
Thank you, Ethan. Okay, the next company up is Sonalisense out of Berkeley, California. Mark.
Thank you. Thanks everyone for sticking around. Thanks to RESI for inviting me. I'm Mark D'Souza. I'm going to be talking about Sonalisense, which is a drug-device combination company. It's a little different. We are phase 2 ready. And it's a little bit futuristic. I don't know if there are any Star Trek fans here, but I think season 4, McCoy says something about surgery. Who needs surgery? And that's pretty much what we're going to talk about today. It's a bit futuristic. We are talking about developing a non-invasive drug-device combination to treat glioblastoma multiforme, a deadly cancer. And really, we see this as a platform technology that could be extended to other cancers. Our vision, which was shared by Dr. Berger, who's the head of neurosurgery here at UCSF, is to turn glioblastoma from a lethal disease into a chronic manageable illness, a pretty lofty goal. So you probably know about glioblastoma. It's a fatal cancer. It's incurable. The cancer always recurs. The current standard of care is surgery, radiation, and chemotherapy. It's a rare disease, an orphan disease, and prognosis is poor. The cancer always recurs. What Sonalisense is doing, as you can see in our tagline, is to develop non-invasive sonodynamic therapy, a word we coined, that combines 2 FDA-approved technologies. A drug, aminolevulinic acid, or ALA, and that targets the tumor. It's needed by the tumor. You can think of it as metabolic targeting. And we combine that with MRI-guided focused ultrasound. And the ultrasound is obviously the device. We have an experienced team. We're phase 2 ready. We're seeking $5 million in a Series A preferred financing. And my partner is Stuart Marcus, who's an oncologist and scientist. He's developed multiple FDA-approved drug-device combination therapies using a process called photodynamic therapy, or PDT. And we've used PDT to apply those principles to sonodynamic therapy. And I'll talk about that today. I'm an entrepreneur. I've been founding CEO of 6 different companies, including companies backed by Third Rock Ventures, Venbio, F Prime Capital, and Nanfung Capital. I've had multiple successful exits, mainly in the rare disease space. We're on track to begin our phase 2 trial in the 3rd quarter of this year with proof of concept about 9 months later. So sonodynamic therapy, or SDT, as I said, uses a device, MRI-focused ultrasound, in combination with our drug, ALA, and we have 3 independent labs that have shown in 3 animal models that you can regress the brain tumors in the models and greatly extend survival in the animal models. And based on those results, the Catherine and Ben Ivy Foundation has agreed to give us $5 million in non-dilutive financing to fully fund the phase 2 clinical trial that will be run at the Ivy Brain Tumor Center in Phoenix by a neurosurgeon named Dr. Nida Sinai. So we're looking to raise another $5 million to support manufacturing, G&A, and regulatory activities. So let me talk about the drug and then about the device. The drug is ALA. It's a drug that's been known for decades. Here, it's used as an adjunct to surgery to image a glioblastoma. So the pink color you see at the bottom is when you expose ALA in the tumor to blue light, it fluoresces pink. And that allows the surgeon to excise the tumor. ALA is part of the heme biosynthetic pathway. Tumors need it. And when the tumor takes it up and you expose it to blue light, it fluoresces. What it also does is that when you expose the tumor to blue light, the blue light activates ALA and it gives off a singlet oxygen, a reactive oxygen species, and that kills the tumor. It induces apoptosis as well. The fluorescent molecule is called protoporphyrin, and that's the principle, a very well-studied mechanism of action of killing the tumor. It's obviously challenging to get light into the brain to activate it, and what we've done is shown that ultrasound can activate the drug. A group in Germany actually did a study where they put fiber optic cables into somebody's glioblastoma, actually into 6 patients' glioblastomas, and actually had positive results. They showed that you can kill tumors with blue light or photodynamic therapy with ALA, but obviously people can't walk around with fiber optic cables in their heads. So while it was great proof of concept, it's not really commercially viable. What we are doing is treating patients with ALA and ultrasound, and using the ultrasound to activate the drug. And what's really cool is that the device, which is MRI-guided focused ultrasound, is FDA approved for essential tremor and Parkinsonian tremors. The drug is already approved as an imaging agent, so we think we can go straight into phase 2. What's really a bit miraculous is that focused ultrasound, if you expose a beaker of water to focused ultrasound, it actually causes sonoluminescence. It actually gives a blue light. And that blue light is exactly the same spectrum, same wavelength as a blue light in photodynamic therapy. So the device is currently approved as to thermally ablate part of the brain to stop essential tremors. We are using non-thermal energies, and we've shown that those lower energies actually activate ALA. And we published a paper in Nature Scientific Reports this year, or last year, that shows that you can get dramatic tumor reduction, that's shown in the top graph there, with focused ultrasound compared to controls, and only in combination with ALA. ALA alone, focused ultrasound alone, didn't do anything. At the bottom, you see extension of survival in rats treated with a rat glioma model treated with ALA. And in the paper, there are very nice MRI images showing dramatic tumor reduction, 70 to 90% tumor reduction after a single treatment. So imagine if you or a friend or someone you know has glioblastoma and they have, wow, you have a year to live, you have a year for a tumor to come back, and you could say, we can treat you with a non-invasive approach. It's safe, already shown to be safe, the drug and the device. No reason you couldn't treat people multiple times. We don't know right now how effective this will be in the clinic. That's what we're seeking the financing for. So we're on track to start a clinical trial in the third quarter of this year at the Ivy Brain Tumor Center. This will really translate the promising data from the 3 rat models into humans. And this just shows you our timeline that both Stuart and I have put money into the company. We've got a few commitments for about $1 million. We need to raise another 4. We're doing it at 10 million pre. We got 5 in non-dilutive financing. We think 5 for IP and know-how we have merits of 10 million pre. So that's really the timeline. We're on track to meet with the FDA in the second quarter and file our IND and start the trial thereafter. So to summarize, we've got a pretty unique technology. It's a drug-device combination. To our knowledge, it's really the first non-invasive therapy for treating brain cancer. There are actually papers that are published on the non-clinical data from the 3 animal models that support us. This is why the Ivy Brain Tumor Center agreed to finance us non-dilutively. We have a team that's done this before. Stuart was Chief Medical Officer of DUSA that developed photodynamic therapy, and several members of his team there are working with us. And this is clearly a big market with an unmet medical need. What I didn't talk about is that this is really a platform. There are papers published on photodynamic therapy in pancreatic cancer, in metastatic brain tumors. So we see glioblastoma as proof of concept that will lead to other indications to follow. All right, thank you. Any questions?
Can you also talk about the barriers to entry, that both things are actually approved and available?
Yeah, so the key to this was really the device. There's a huge patent estate covering focused ultrasound. As you might imagine, you can't just take your normal ultrasound and put it into somebody's skull, expose somebody's brain to it. So Inside Tech, who's developed this technology, has a large patent estate in this space, and we are negotiating an exclusive license with them. That will be a closing condition to the financing. We hope we'll do that fairly soon. And we filed an application on the combination of ALA and ultrasound. We've also come up with a proprietary formulation of ALA. ALA is currently approved as an oral drug. It's subject to first-pass metabolism, so you have all sorts of tox issues with it. So we think we can get some claims issued on our IV formulation. And it's really the combination of the two. And drug device development is a pretty unique skill set. Not everybody, I think, can easily jump to it.